In this video, we're going to talk about ATP synthase and its role in ATP formation. So we've got two synthase enzymes depicted here, one on the right for mitochondria, and one on the left to depict chloroplast ATP synthase. So each ATP synthase is made up of two particles. There is a sub-zero particle, which in mitochondria we call F sub-zero, and there is an F1 particle in mitochondria. And in chloroplast, we refer to the particles as CF sub-zero and CF sub-1. So your F sub-zero particles will be the ones that are actually embedded into a membrane. So in your chloroplast, this is the thylakoid membrane, or you might see it referred to as the internal membrane on your PowerPoint. And on your mitochondria, you will this ATP synthase F sub-zero head is embedded into the inner membrane of the mitochondria. So in each synthase enzymes, the sub-zero head is going to be responsible for being directly rotated by protons. So our F sub-zero heads are going to see a large concentration of protons on the side closest to them. So on the chloroplast, this would be on this side, and in the mitochondria, it would be on this side. So thinking back to the electron transport chain or the light-dependent reactions in photosynthesis, we know that we were causing a proton gradient. So in mitochondria, the proton gradient, we were pumping out protons into the intermembrane space of the mitochondria. So this area with higher protons will be the intermembrane space. And in our chloroplast, we know that we were bringing in protons into the lumen of the thylakoid, thylakoid is a part of our chloroplast. So the inside here with the higher proton concentration will be the lumen, and the outside will be the stroma. So our sub-zero particles are going to be the ones directly interacting with protons, and our F1 particles are going to be the ones that are going to be the catalytic sites for the formation of ATP. So in the mitochondria, the protons are going to travel through our F sub-zero particle, and they're going to travel into the matrix. And in our chloroplast, the protons will be traveling from the lumen into the stroma. So as these protons travel through our F sub-zero particles, they're going to be rotating C subunits. So C subunits are just subunits of our ATP synthase that form a ring, and these protons will travel through this ring, rotating the ring. And then these protons will leave out of a channel that will take them into the opposite compartment. So this blue part of our F sub-zero particles are the C subunits. So the whole reason for causing this rotation is to cause rotations in this stalk that is connecting our F sub-zero particle and our F1 particle. So this stalk in both is referred to as the gamma subunit, or the gamma stalk. So as our F0 particles are rotating, it is going to be causing rotations in our gamma stalk, which is going to cause conformational changes in the proteins of our, or the protein subunits of our F1 catalytic head. So these conformational changes are going to cause the actual catalytic reaction of ATP formation. So this driving proton force is referred to as proton motive force. It could also be referred to as an electrochemical gradient, and it is also called a pH gradient. Because with a higher concentration of protons, we have a lower pH, making that compartment more acidic. So on average, in any living cell, you're going to see a much more acidic lumen and intermembrane space compared to the stroma or the matrix. So since the rotation of these C subunits rotating the gamma stalk, which will rotate the F1 particle and cause conformational changes, this whole scheme of catalyzing ATP by rotations is called rotational catalysis. So our F1 particle is made up of two different subunits. We have a beta subunit and an alpha subunit. So the beta subunit is the actual catalytic site for our ATP formation. So if we did a cross-section of the F1 particle, it would look a lot like this. So this middle stalk would be our gamma stalk. And we have our three beta subunits, our three catalytic sites here. So there are three different conformations a site can be in, and each one will be in this site, or in one of these sites at any given time. So we have the open conformation, the loose conformation, and the tight conformation. So this will go in a cycle. So it'll go open, loose, tight, then open again. So it would be a cycle. So the way this works is, depending on the conformation of the site, there will be a change in affinity for ADP and an inorganic phosphate. So if a site is open, it will have just given off a completed molecule of ATP, and it will be ready to pick up another pair of ADP and inorganic phosphate. So if it's open, it's just given off an ATP molecule, and it's ready to pick up a new ADP and inorganic phosphate. So that is open. So if a site is in loose conformation, it will be holding ADP and inorganic phosphate closely to it, waiting for the reaction to occur. So this conformation is going to have a really high affinity for ADP and inorganic phosphate. So this is loose. And the third conformation is tight. And this is when the greatest affinity for ADP and inorganic phosphate will occur. So at this point, ADP and inorganic phosphate will be so close that the spontaneous reaction of ATP formation will occur, and this molecule of ATP will have spontaneously reacted to create the molecule. So ATP synthase doesn't actually make the molecule, it just has such a strong affinity at a certain point that this inorganic phosphate and ADP molecule are so close that the spontaneous reaction does occur. So remember, spontaneous negative delta G does not need energy put in for the reaction to occur. So ATP synthase's job is just to get them so close together that the spontaneous reaction does occur. So you can imagine this like a carousel. As your gamma subunit rotates, it changes the conformation of each one of these sites. So our first open site, when this gamma subunit rotates a certain amount, which is 120 degrees, this open site will then become loose. And if we have another 120 degrees rotation, this loose site will become tight. And if we have another 120 degree rotation, our site that was originally open will now release its new ATP molecule that it formed. So that's why we say for every 360 degrees, for every one full turn, we create 3 molecules of ATP. Because every site has gone through all 3 conformations and released 1 molecule of ATP. Right? And again, this is our rotational catalysis. So just a brief overview. This whole reaction occurring it's a chemical gradient, so chemical energy to mechanical energy by this physical rotation, and then stored again in chemical energy. So the first one would be more electrochemical energy. Sorry about that. So electrochemical energy to mechanical energy by actual rotation of the C subunits, rotation of the gamma stalk, which causes these conformational changes in our F1 subunits, which causes chemical energy in the form of ATP. And just to clarify, the F1 particle is not physically rotating, it's the C subunits rotating, so it's the stalk rotating. So when I say 120 degrees, I mean 120 degrees of the C subunit rotating the stalk and causing these conformational changes. Our F1 beta subunits stay put. So in our mitochondria, we'll be releasing these ATP molecules into the matrix, and in the chloroplast, we'll be releasing these ATP molecules into the stroma. From there, the ATP molecules will be used in the Calvin cycle.